Smart Foodscapes annual advisory board meeting. Thank you for being here. We know the weather has been a little bit of a problem today, so I appreciate all of you coming despite the weather and at this time of day. And then I think we have also some presenters and people on Zoom. And just, to give the introduction, I think what we'll do is people that we had a dinner yesterday that didn't introduce themselves will have the opportunity to do so today during the meeting. Yeah, let's see if not. Yeah, the next slide. Just, to start this meeting. Yeah, this. Okay, there it is. So just a brief overview of the plan that what we want to accomplish today with this really short presentation, just to engage people into discussion for the afternoon brainstorming sessions. Then the program for tomorrow is going to be just reporting this, what came out from those sessions, those discussions, and then hopefully we can, develop like a roadmap for just short-term objectives on this project and also long-term, either with things that we are planning on accomplishing in 2023 and then beyond. And that's, going to present just my story in a few minutes, this idea that it's been challenging, but I think it's working for us, this keep this integrating fluidity in between the extension, research, and education. I think this is a really important part that I think NIFA acknowledges the importance of these interactions and not doing just research or doing just education or extension, but trying to integrate all this into a true, living organism. I think that's what I like to see. And then that we all get interested, not just on the research or the extension, but also on the education, and people in the education getting interested in research and extension. And my story today is just, with the issues that we are facing on crisis on the beef industry, for instance. And then I was in Europe recently, and this idea of reductions in the supply of beef for the population, some agendas for some groups that tend to reduce all these animal husbandry activities to a point that some are predicting that some countries actually want to just eliminate agriculture all of a sudden. And I don't know if you know, I follow this Frederick Leroy, this scientist and professor, that he, makes a really good point on what meat means to us or meant to us in the past, and then for the majority of people. But then we have a loud minority that, think that or are proposing that meat is a different story. And then, well, livestock is blamed for many things, from cancers to problems on environmental impacts. And as he comments in that that presentation that is also online, is basically based on bad science, or scientists essentially focusing on bad science. Even some scientists that from the human nutrition perspective are good. So I think that's an opportunity for this program to not just only develop good science, but also good extension and education programs that, highlight like these people that they are working in the field. Like, this is on this picture, this is the mother of a producer I met in Spain. She's, 80 years old, and she every day herds sheep back to, pens them overnight, protecting them from attacks from dogs and wolves. And then, she's thinking on keeping that land for the future generations to come, including his son, and then his all the descendants of this generation. And then, trying to keep the soil, keep the health of the soil, the health of the animals, and thinking on the health of the population that consumes these products. So I think that through this fluidity of extension, research, and education, we can accomplish all these things, not only just doing the good science, but also trying to extend and educate our next generations of land managers and producers and policymakers that the importance, of these people that are working and then preserving the integrity of our ecosystems. So that's, I think if this project, puts like a small brick into this building, I think it's going to be a really significant accomplishment of this project, right? So it's doing the good science, a good extension and education. So, and then with that, we can start, this series of presentations or espresso presentations. And the first one is going to be Jennifer McAdam. So she's been working on plant, she's forage plant physiologist and forage specialist, and she's been working on the species that the identity of the species that we need to incorporate in our islands of diversity. So I think we can, it's already on.
Okay, thanks, Juan. It just makes one hand free anyway. And let's see. Is the slide advanced? Sorry. No. Can you advance the slide? Okay, so this is one replication of one of our sites. And I'm sorry, I can't see my own slide, but we're starting with objective 1A. And so that's to screen plant materials for the resource islands. And so, sorry, can you go back a slide? Okay, so I don't know if it's clear, but you can see that there are some blue colors on there and some pink colors, perhaps on the lower part of that slide, and a little green strip at the bottom. So we have a number of legumes, which of course are beneficial because they produce their own nitrogen. And then we have a number of non-legume forbs that we'll just call forbs. And so we have 4 sites altogether, but 3 under this grant where we are looking at these and trying to establish them at an optimal time. And so our goals are to have plant materials that are not only productive, but are well adapted, of course, that are going to provide high quality for the ruminants, and that will also have at least one bioactive chemical like tannins. I've done a lot of work with tannins, and so we know that they can have benefits. We have plants like small burnet that produce hydrolyzable tannins that have a little more severe effect on the animal, but have some really good effects on maintaining nitrogen in the feces instead of in the urine where it gets volatilized. And so all sorts of potential health benefits for the animals, like improved productivity, as well as environmental benefits. Next slide, thank you. So this shows our 3 sites under the grant, one in Logan, one in Ephraim in central Utah, and one in Cedar City in the southwest part of the state. And so what we did, just to keep some confounding effects out, was to do soil testing in all the locations. And mostly what we added was either nitrogen or zinc or iron, sorry, not nitrogen, sulfur or zinc or iron. So those are, a couple of them are micronutrients that were deficient, so that we didn't have that confounding factor. And then we added mycorrhizae. Mycorrhizae are soil, it's a soil fungus root association that is beneficial in acquiring nutrients, especially phosphorus, which can be especially important to legumes. We want these legumes to become deep rooted because they're on dry land. Next slide. And I'm just guessing at what's on these slides. So this shows the precipitation at our various locations. We had a site that we established under the seed grant that Juan got preliminary to getting the CAP grant. That is at Clarkston, and the growing season precipitation, which was from April 1st until the end of October, 2.41 inches at Clarkston. And so I was surprised anything came up because I've been working with irrigated forages for most of my career. And then you can see the rainfall amounts, I hope, at Logan, at Ephraim, which also was about twice as much, but low, but in Cedar City as well during the growing season. Clarkston doesn't have any irrigation available, but on the advice of an advisory board member, the other 3 sites were selected so that we can apply irrigation, because we only have a short window to get the information that we're going to get from this grant, and hopefully maybe go on to have information that would get a second follow-up grant. So our objective. The other objective of this is to supply dry matter for the ruminant nutritionist so that she can do in vitro studies at least. And we didn't harvest any plant material this year, but what I had done was establish plants in the greenhouse as verification plants so that I would at least, some of the plants I hadn't worked with before, so that I would at least understand what I was seeing in the field. And so those plant materials I've been collecting, and they're definitely not being cultivated under the same circumstances, but we hope that we can get information on the potential for them to produce phytochemicals and so on. So next slide. Hello? Thanks. So I did collect a little bit of information this summer on the plant materials at Clarkston. And so you can see the species that were most productive. And these data are in the number of plants on a per acre basis. So these aren't pounds or kilograms or anything, but the plants that we saw in the spring last year or early summer, Sandfoin, small brunette, Ladak alfalfa, those are the ones that we pretty much assumed would be productive. And then Utah sweet vetch is a native, purple prairie clover is a U.S. native, bird's foot trefoil is another introduced species. So you can see the ones that we could find in August, and then by October we had some additional plants. We had blanket flower came up, showy goldeneye came up, and looks very well established at Clarkston. So it's a whole new area of plant physiology for me to see these plants that can apparently put down roots and germinate and put up huge plants on almost no water whatsoever. But we are finding some success, some usual suspects as well as some very nice natives. So I think that's my last slide. If you want to do one more. Thank you. Yes, sir. I don't think I had it on the slides. So the time of year we were planting, we planted initially at Clarkston, I think it was the 19th of November, just before really cold weather set in. And I think I showed a slide actually at the annual meeting last year of that plant material going in. And then in the spring, by May, we didn't see anything. And so we replanted at the beginning of May. And showy goldeneye, for one, was, I know, not planted until the spring. So because we put everything back in the same plots, except for a few new species that we traded in, we couldn't say for sure, but I suspect we didn't have much success with our late fall planting, which I thought was the perfect time. I thought we got it in just at the right time and snow started coming and so on. Yep. Yeah, I think I'm remembering correctly, RTI. And I got it through Granite Seed and they had it in Denver. And so we've had it shipped here. It comes looking like it's cat litter. I think basically it's on clay. And so we incorporate it, and we've been incorporating it along with the nutrients, the mycorrhizae. So I hope that we were a little concerned. We weren't sure if we could mix it without it coming to some harm, by association with the sulfur and zinc and iron, but that's how we applied it. We haven't, but I have a colleague who has done a fair bit of culturing of mycorrhizae. And I know at Clarkston, we applied it to the center of the plots. We weren't absolutely certain from GPS how large the entire plot area was. And so we know that on the edges, we didn't apply it, and in the center we did. And so my plan is the only native I can think of right now was Utah sweet vetch that became established earlier, but next year we are planning to compare plants from the perimeter with plants from the center of the plots to see if there's a difference in the mycorrhizae. Oh, sorry, I forgot about recording. So the question was if we had tested to see if the natives had been infected. Is that, maybe I didn't even think about your question deeply enough.
We're gonna take just a few-minute break because we're gonna have a monitor put up here so the presenters can see their slides. So take a minute to point that and then go back to the monitor so that we'll continue with our next presenter.
Well, I did remember I was supposed to ask the audience for, their input on certain things. And so, okay, just my one sort of blanket question is how much should we interfere with these plots? Should we just put them out and then, except for noxious weeds, they're on their own, or should we go in with herbicides? Are we wrong to try to bring the fertility up? How much, since what we're talking about are discrete islands on rangeland, what would be a realistic amount of inputs in those areas? So the comment, I guess, is should we just basically let nature take its course? And then intervene if necessary. And I think the only thing I would say in terms of these all have 4 or 5 replications. And then I think we're hoping that we can at least discuss them in the same paper. And can we do that if we do one thing at one place? It seems like we kind of have to have a policy starting out. Yeah. So for the recording, that would be more what a producer would do, would be to intervene if necessary. Yeah. So an economic valuation would be one way to look at it. Juan, could we just get the microscope the microphone for the questioners? Then you can use your own words and I won't repeat them.
Jennifer, while they're getting the other microphone, there's a question from Zoom. And the question is, are you using GIS to locate the plots for the Foodscapes? And if so, how?
So to answer that question, no, we located our plots on land that either USU controlled or a cooperator controlled or collaborator controlled, and that had irrigation available. So did you want to finish your
Sure,
comments?
I guess. So I think there's different ways to put dollar value behind it. And one is by talking to producers of what is the forage value, or whoever, can provide that information. Another option would be looking at cost share programs that are available to ranchers and looking at what cost share do they provide for different range planting types. And then kind of working backwards from that. So I would start at that level, but then obviously for the project, you need some level of success out of the plantings for the additional information. And so I think, like you said, if you need to intervene with irrigation because it is an incredibly low rainfall year, then do that, but just acknowledge that we met the economic threshold for what a producer might do. And now we're going to this additional threshold for the value of the information, the research information. So that's, I would kind of tier it like that. And even, having a set of criteria of the soil health is probably like one item, irrigation is another item, spraying herbicide is another item. And again, tier it based on what you think is going to produce that outcome, the desired outcome.
And the only thing I'll add to that is that we are taking care to keep track of, any additional labor, any additional inputs and so on.
What I'd add to your question too, or about intervening is, now I don't know, other people probably may have had different experiences, but my experience with planting was up in Rich County at probably 6,500 to 6,800 feet. And we did about, 3 to 5 different large plantings where we did part of it in the fall and we never finished, and part of it in the spring. And in every instance, the part that we planted in the spring was a forb plot. And what we planted in the fall was grass with less than 1% forb. I mean, it was the same seed mix in the fall and the spring. And most of the forbs were Ladakh or Ranger alfalfa, sainfoin, small Burnett. And in some cases, we had some Sicer milk veg. But in every instance, the spring part of it was forbs and the fall part of it was grasses. Just so one thing you might think about if you're doing replicates is to just plan to do some of the replicates in the spring. And it might kind of help answer that question too, if you get a difference. The only thing that I ever heard that would partly explain why we saw what we saw is that alfalfa seeds have a bad habit of, if the temperature gets up to 35 degrees one day, they'll break dormancy, and then it, goes 20 below again and kills them.
Well, that's what we suspect happened to our fall planting, was just that it warmed up enough. And we have a weed scientist who's done a lot of rangeland work in our department. And he said, I get so mad because everybody says you have, plant in the late fall, plant in the late fall. And it never works. And so I think that our one experience at Clarkston would definitely be in line with what you just said too, because our replanting, we got the usual suspects and those great, Sandfoin and so on, small Burnett.
Great, great discussion. Yes, so we can continue on the brainstorming sessions on that. So we keep, we'll bring eventually the other monitor, but I think we'll have another. Yeah, thank you, Jennifer. So our next presenter is Doug Ramsey. So it's a little bit of change of a program, but we want to show now our GIS specialist, the work he did on spatial arrangement of the island. So that's the critical point of where to put these resource, legumes and forbs in the landscape, and that's a really big headache.
All right, thank you. The reason Juan was nice enough to move me up a little bit on the schedule is that I'm faced with a once in a career event where I have to give 2 different papers at 2 different conferences in 2 geographic locations within half an hour. So I got to run to my office and get on Zoom and give a paper in Salt Lake. So if I can have the first slide, or maybe I can get this to work. Oh, yeah. That's the back, that's the forward. All right, so my task this year was to identify the best locations to place these islands so that they have the plants that they seed in these islands will have the best opportunity to germinate and grow. And the first thought that came to my mind was, in an environment like ours, is a water limiting environment. That water will be the primary driver of where things could grow more easily. Other things like soil type, soil depth, slope, land use types within larger pastures might affect it. But in this particular case, this test case in Richmond, where we have this initial pasture, everything seems to be fairly uniform as far as terrain is concerned and soils. Soils vary a little bit, but the limiting factor would just be water here. So we're just focusing on the water aspect of it. So the pasture is about 53 acres in size. We first broke it up into these 6 pastures, just using a simple GIS function to break up a larger polygon into smaller polygons that were even in size. There's no attempt to make the pasture shapes fancy or fit a certain parameter of the landscape, but they're just basic rectangles. And each ended up to be about 8.9 acres in size for these 6 pastures. 3 of them were identified as the treatment pastures, I think randomly, which will be a problem later on, we'll see, where the real world hits the modeling world. And the other 3, the ones that are not in red, were the control. So there'll be no islands in the control pastures. There'll be 3 islands in the treatment pastures. So in order to identify the areas that have potentially the best opportunity for these plants to grow, I settled on using remote sensing imagery. So the better half of geospatial sciences, the other half is GIS. So there's my bias. So using remote sensing, I thought I could use the level of greenness for any given growing season and identify those pixels that remain green or are green or greener than other pixels in that same pasture year after year. So I used Google Earth Engine to do this with, because it's really easy to access a historic data set of any place on the surface of the Earth. In this case, I used a European satellite called Sentinel-2, which is almost perfect for this scale. It's not quite as resolute as we want it, but it's as best we can get for those 6 years. I sampled across 6 years from 2016 to 2022. And for each of those, I calculated the greenness values for every pixel, calculated the means, generated standard deviations and selected all those pixels that are greater than 1 standard deviation above the mean of greenness. And this kind of is a better depiction of what's going on. We have the raw imagery on the top left. We generate the NDVIs for each one of those images across each one of those years. These are, by the way, July and August images. I picked those purposefully because when I monitor landscapes like this and I go for spring imagery, there's so much variability depending on the water year that it really fluctuates. You really can't tell what's going on. If you pick later summer, everything is green and starting to senesce maybe, but if they're going to be green, they're going to be green at that time of year. Everything's going to be green in spring. So these are all mid to late summer images. From that NDVI image, the greenness image, I calculated the mean of the entire pasture and the standard deviation of the entire pasture, and then identified those pixels that were greater than 1 standard deviation above the mean. I chose 1 because it was the first one. I could have chosen 2, but standard deviations, but we would have received far fewer pixels. From that, I just counted up the number of times for each one of those years. Those pixels became green and came up with that image on the bottom left. So where red represents pixels that were green for only 1 year out of those 6, and the greener pixels are green for 5 to 6 years out of that set. It was complicated a little bit because that line of green on that east side is adjacent to an orchard which is irrigated, so it kind of throws it off a little bit. This was the final map that we went from, and to identify where these islands are to go, we try to match up the islands to the greenest areas of the pasture or the individual paddocks. And that's where we hit a slight snag because sometimes the greener pixels occurred in one corner and not in another. We want to distribute these as much as possible. So reality isn't as nice as the modeling world. So we ended up with this. The blue areas are the pastures or the islands, distributed as best as we could distribute them given the distribution of greenness within each one of the paddocks. And you also notice that some of those islands cross the fence line. That's because our sighting of the pastures on the field wasn't as exact as it should be. 2, when it was plowed and then seeded, the GPS on the vehicle that plowed the lines was also a little bit off. So we ended up with these semi-rectangular areas that are fairly uniform in size, but different shapes, which should be okay. We just have to move this fence a little bit farther south and this one a bit farther west, and we should be okay. From this point on, we will, as of now, the islands have been cut in. They've been plowed and seeded, correct? They've been seeded. And I guess we'll wait and see how they grow. This pasture is a non-irrigated grassland pasture. So it's fairly uniform in area, in type of growth form. It's heavily managed by Utah State to produce hay. And so we'll see what happens with the islands. From here on in, as they're populated with cattle, we'll figure out ways of tracking the cattle either through GPS, if that's going to be accurate enough in such a small area, or cameras to identify movement patterns and where the cattle are going and where they're loitering. I think that's it. Any questions?
For asking the questions.
Are you, by water, you mean, I guess I need to talk into this thing. So by water, you mean the soil moisture, or are you talking about the watering points? Okay, so the watering points are going to be located along that east to west long axis, mostly because of access, and as far away from the islands as possible. So we'll maximize the distance from the islands for the watering points.
And I'm just thinking about how you'd actually look at GPS points with a GPS count on it. It'll look like this big block in such a small.
That's what we're worried about. So it's, we either use those or we can use cameras to monitor the site and then physically map where the animals are. So the GPS collars, I think, will be a little too coarse. I guess it depends on whether we can differentially correct the points or not.
I wonder if you could break out the points within a certain amount of time and then just summarize the difference.
So, yeah,
I do it on a per day basis and then look at the distribution faster.
no, so just on, in certain times, go and actually monitor where the cows are. Yeah, you might have to do that. Might be the best way to do it in such a small area.
On big huge pastures with collars that I've done, it's still a big blob on huge.
Yeah.
The challenge on a tiny.
Yeah, that's what we're trying to figure out. All right. Oh, go ahead. One can speak better to that, but they were plowed, and they were sprayed, plowed and then seeded. Is that correct? One, how the sites were prepared?
Well, the question was, yes, you can.
So I was just wondering about the process for getting the seeding in, how you prep the ground and then as well, time of year on that, spring seeding, fall seeding.
Yeah, so it was spraying. So we spray the plots once in the spring and once in the fall. And then that was mostly brome grass. And then is non-till seeding. And the seeding was in November 17th. The seeding of all the species. I want to show the plan, how we did the different strips of seeding. Yeah. That's, so the idea for that is something we want to discuss too, is try to conserve moisture on the soil with no tilling. Yeah. Any other questions or? And
So, how do you think elevation will become a factor when you're applying this on a site that's not as flat as this? Where do you think?
I guess it depends on the difference in elevation. I mean, if we're looking at a few hundred feet change, that might be a problem. Most of that will probably have to do with slope and how water comes off the surface and exposure. So I suspect steeper areas, I have shallower soils and they'll be less equipped to allow that growth. Let's say a flatland. The other problem is pulling out wetlands and riparian areas from a larger pasture. So in a larger pasture, you have to throw in more variables. This one is just relatively flat, gently sloping plowed field, basically.
Yeah, I imagine when you apply that on a broader landscape, it's going to focus more.
You would have to add more variables.
drainages and things like that.
Yeah.
Thanks.
any more questions? Yes, so we just. Oh, okay, sure.
Both in the seeding methodology as well as just monitoring moisture on landscapes of, so methodology, seeding depth, was it uniform for all species? It was a no-till system. So I guess this is a question for you. Was it all uniform depth, and what depth was seeding? Do you know, Juan? Oh, well, was it like a grass seeding of like ½ inch, or was it shallow depth seeding for like 1/8 inch, basically? Okay. Okay, just because there is variability, a lot of variability in some of these more native species that don't like being planted that deep. But then the question for kind of the GIS side of it as well is microtopography. And you went a no-till system, but you know, in a lot of systems we're finding roughing that surface, not having just a ag field really benefits these native species in particular in establishment, and just considerations for, you know, monitoring or looking at some of that microtopography and moisture capture especially in the establishment phase.
Yeah, if you look at that image on the screen, the yellowish to greenish pixels are all following drainages. So you really can't, when you're in the field, you can pick out the drainage, but it's been plowed over so many times that they've flattened that a little bit. But you can really pick them out of the imagery and in the LiDAR data that we have for the site. So the drainage is basically driving the moisture availability.
Great. Yeah, thank you, Doug. So our next presenter is going to be virtual. So I guess I need to put her in screen is Fernanda Batistel. So she's our nutritionist working at University of Florida. And she's going to be talking about how we can look at these interactions between the range vegetation and the islands from, you know, bottom up. So trying to see that in the field, in the lab, and then into the field. Yeah, Fernanda, good morning.
Hi, good morning. Can you guys hear me okay?
Yeah.
Okay. Do you want me to share my slides here, or are you planning to share over there?
I think you can share the.
I'm getting a message that I'm disabled to do that.
I think you should be able to do it
now.
now.
Yeah, now I got. Thank you. Okay, so I will be responsible for objective 1C of the project. So it's pretty much a follow-up section from what Jennifer will be doing. So she will be collecting the forage samples there. She will be sending to us here. So I'm working together with Taylor Jackson, that is my master student, and we'll be evaluating these forages over here. So the objective that we have on the project is to evaluate the effect of these specific forages and also their combinations on rumen fermentation, microbial growth, and nutrient digestibility. And we'll be using what we call continuous culture fermenters. That is the picture that I have here. So this illustrates the system. And I will give you more details in the next slides how this system works. So besides the objective that we have in the project there, before discussing with Erwan and Jennifer, we decided to do a pre-test there, or another small study before the one using the continuous culture fermenters. So in this pre-test, what we'll be doing is to use a batch culture system that is a very straightforward system. It's just small serum bottles where we can put the samples, put the grooming fluid there, and then we can see the production of short-chain fatty acids that are a very important source of energy for the animals. We can also see methane production and CO2. So the plan is to do this batch culture study early in the next spring of 2023. So Jennifer already collected the samples, and I just needed to figure out with you guys the best combination to using the batch culture system. So again, we are planning to do this early next spring with the samples that Jennifer already have there. So if you are not familiar with the continuous culture fermenters, that is our official objective in the project, is this system here that mimics the rumen or the main stomach of a ruminant animal. So this is very straightforward system where we can put the samples inside. We'll be feeding this jar here twice a day with the forage samples. We'll be putting the rumen samples here, rumen fluid here, and we can keep for this fermentation going on for several days here. We will be collecting the samples that live here, so we call the outflow line here. So this mimics what is leaving the rumen of the animal and what is getting in the small intestine, pretty much. So we'll be getting the samples here to analyze nutrient digestibility. We also will be getting samples to analyze the short-chain fatty acid production, the methane production, and microbial growth here. In this system here also will be collecting pH data, so the variation throughout the day. And as we can see here in the details, this system mimics very well the rumen, except for absorption of nutrients. So we have delivery of a buffer that mimics the saliva production in an animal. We have a CO2 port here that will be keeping the environment anaerobic, because in the rumen of the animals, we don't have oxygen there. We also will be keeping the temperature that is 39 degrees Celsius, that is the body temperature of the animals there. So the experimental design that we have in the grant proposal is a 8x8 Latin square design, and the periods will be 10 days long. This means, excuse me, this means that we will be adapting the fermenters during 6 days. We'll be just be feeding them and taking care of the pH in this period. And then in the last 4 days of the experimental periods is when we'll be collecting the outflow samples here and also the samples from the fermenters to analyze. So we have 8 replications of the treatments there, and we'll have a total of 64 observations from this study here. For the treatments, what we planned before is to have a grass as the basal diet or the control diet. And then the remaining treatments, the remaining 7 treatments will be a combination of the basal diet that will be around the 85% of the treatments, plus 15% of the dry matter will be combinations of legumes and forbs species there. So we'll be arranging these based on the samples that Jennifer will be collecting. And here is a summary of what we are planning to analyze. So the short-chain fatty acids are, as I mentioned before, are very important for ruminants because they can provide up to 75–80% of the energy requirements of the animals. So it's a very basic analysis that we do when we analyze rooming fermentation. We also will be analyzing ammonia count nitrogen that will be indicating protein digestion or protein fermentation in the rooming of the animals. And then another key thing that we'll be analyzing will be the microbial growth here. So the microbial growth here is key because once the microbes, they die and leave the rooming, or because of the high passage rate of the rooming, they will be ending up in the small intestine. They will be digested and they will be used as an amino acid source for the ruminant animal. So microbial protein here is the best protein source that we can have in the diet of a ruminant. So if our treatments, our forages can increase this is a very good thing. We'll be analyzing the nutrient digestibility here of the treatments. So we have organic matter, fiber, protein, starch, and specific fatty acids there. And also we'll be analyzing the total gas production during the fermentation. And we also will be analyzing methane specifically here because as I'm sure you heard before, this is related to the environmental impact of ruminant animals to the environment. So is a measurement that we consider important here as well. And so pretty much that was my presentation. I don't have a lot of results to share yet, but this is what we have planned here. So if you have any questions, please let me know. And if you have suggestions to improve also, I will be very happy to hear your comments to improve what we are planning to do here.
Yeah, thank you, Fernanda. So any questions? Yeah.
Fernanda, can you hear me?
Yes, I can, Jennifer.
So the question that I would have, since I've worked with tannins and we talk about tannins precipitating proteins in the rumen and letting them also leave and be digested later on in the animal's gut. Do you know how different or comparable the plant protein is to the microbial protein? Are there big differences?
So if we just check the crude protein, will be similar. But if we check the profile of amino acids there, then the increasing the microbial protein is a big advantage there. So if this is true for both, if we think about beef and dairy cows as well. So if we get a muscle sample, a milk sample, and analyze the profile of the amino acids and compare those to the microbial protein, they will be very similar. So that is a big advantage of increasing the microbial protein. I also wanted to say that there is it's good to improve the protein, the dietary protein that is arriving in the small intestine, especially when we think about the high producing animals. And I will use dairy cows as an example because it is my main area of research. But for example, early lactating dairy cows, the microbial protein is not enough to meet their amino acids requirements. So we needed to add what we call rumen undegradable protein there. That is pretty much when the dietary protein escapes rumen fermentation and arrives in the small intestine. So we needed to have both there, microbial protein and the undegradable protein there to meet the animal's requirements there. Did I answer your question, Jennifer?
Yeah, thank you. That's interesting information.
Yes, I have another comment, on the on the brainstorming session, something that you mentioned, Fernanda, in terms of which species, which combinations we could try in these fermenters. And then maybe some with that initial design you showed us, maybe try some additional combinations, but that's
Yeah,
some of the discussion I think would, for the short-term goals, I think will be to, well, which legumes and forbs we need to mix and try to look at the impact of those on all those parameters.
yeah. And if we have like the plant material, definitely we can do a lot of combinations using the batch culture system, because we can analyze the digestibility, we can analyze the gas production and methane over time, so we can have measurements every 10 minutes. And then from that data, then we can refine the plant material that we'll be using for the fermenters there. So I think we have a good plan. We just needed to keep thinking about these and find out the best plant materials to use and combinations to use it there.
Since we have that screening system, will be nice. I think if we can discuss this afternoon potential combinations that we can try, and then given that we can screen different combinations, so that should be something that really curious to
Yeah.
what the benefits are just from the in vitro perspective. Yes, another question.
Thanks. I maybe missed this. I was just curious how, I guess, right, this is in a lab. So, and I'm thinking about the cows out on the pasture or the range. And so does this even matter? But accounting for saliva, mastication, existing inoculation in the gut, how can you, I guess, account for that, or does
Yeah,
it?
Yeah, that is an excellent question. So when we use this system, what we will be doing for the saliva, so we'll be using rate of delivery here, using these lines here that mimics the rumination of a cow. So we can change this. For example, if we wanted to mimic dairy cow, the cow will be producing up to 200 liters of saliva in a day. And then we can, considering the volume that we have inside of each fermenter, and see how much saliva will be delivering throughout the day. For mastication, we cannot really mimic here very well in the system, but what we do, we grind the samples or we make pellets. So we have two options there. I think the best option is to make a pellet there, because it's what the others have been done when they use the system. So when we think about in terms of a publication, that will be easier if we make the pellets. And that I will be able to do here when we have the material for the fermenters. For the inoculation of the microbes here, what we do when we start the period, we go to the farm and we have rumen cannulated animals. And then we can collect from 2 or 3 animals, bring it to the lab, and we keep it like an anaerobic conditions, temperature and everything. And then we mix everything, and then we'll be inoculating the fermenters here. So by doing this, we don't have an effect of the type of the microbes that we have there at the beginning. So all the fermenters will have the same microbes, and then the effects that we'll be seeing there later is because of the diets, not because of the type of microbes that we have there. We may favor like specific forages may favor later a specific type of bacteria or other, but then is a treatment effect. We are removing the confound factor of the inoculation there. Did I answer your question there?
Yes, thank you.
Mm-hmm.
some items to discuss, Fernanda, yeah, for the brainstorming session. But yeah, let's move on. So before now is Pablo Gregorini. I think hopefully he's still awake. He's in New Zealand, so his time is about 4 a.m. I think 4. So he had to wake up a little bit early today. So hopefully you are still awake. And so he's going to present some of the modeling. We'll be working with this virtual cow now instead of, that the in vitro. This is now a cow in the computer that, trying to determine some grazing systems, some grazing pattern that we need to do with these cows in these islands. And so one of the best ways to do this is try to, look at different scenarios with a model. So that's the first talk that Pablo is going to present on the Mindy cow. Pablo, are you?
Now, can you hear me?
Yeah, a little. Yeah, try again.
Can you hear me, Juan?
We can hear you now. Yeah.
Oh, that's good. I can see. Well, I saw myself there a little bit. Hopefully, you can still hear me and see me.
Your slide.
Okay. Can you see my slides, Juan, now?
I can see you now, not
You
your
can see,
slides.
not the slides. Let me. Maybe it may be easier if you got them around there. But let me see. Share. There we go. Let's just bear with me. Okay. How about now? Can you see my screen?
Yeah.
Okay. But well, first of all, thank you very much. Yeah, it's early in the morning here, and so bear with me. And just in a couple of hours, Argentina is gonna play Croatia, so I had to wake up early anyways. The World Cup. So if you can't hear me, please let me know, because my connection where I live is not as good as you may think. I'm Pablo Gregorini. I'm part of this project as well. A very good friend of Juan and Fred, and I also a good friend of Stefan Van Vliet that I've seen in the audience there. Where we are working together as well. I'm the leader of the Lincoln University Pastoral Livestock Production Lab, and I'm the head of a Center of Excellence for the Design of Future Productive Landscapes. So one of the things that we are doing for this project is run a modeling exercise. We are modeling the effect of the frequency and of the allocation and proportion of tannin containing legumes to enhance the performance and reduce environmental impact of cows grazing grass dominated rangelands. So it's a model that I developed in my previous life where I was working at Dairy and Cetice, and Juan and Fred had a lot to do in the development of this model, especially in the last two modules. So this model is called Mindy after my wife. It's a mechanistic and dynamic model. It's a cluster of models, right? And this model represented the diurnal feeding and grazing pattern of a cow, amongst several other things. The model is composed by 5 clusters. One is the modified version of Molly, who was developed in UC Davis a while ago by Lee Baldwin. And then the model has sward canopy structure and herbage quality that simulate differences in sward canopy structure and chemical composition through time, including diurnal patterns of chemical composition of the herbage. It has a model of ingestive behavior, a model of feeding motivation, a model of preference and selection, a model of foraging bioenergetics, and the seventh model, it's a model that represents drinking behavior and urination behavior. This is just to give you a rich picture of the complexity of the model. I'm not proud of being of saying that the model is complex, right? But because it's a very detailed mechanistic model and dynamic based on physics and chemistry, we can answer questions at different levels and ask the question, why did it happen, rather than what could happen if it was a simple predictive model. So on the left side of the screen, you'll see a general representation of the feeding motivation module of the model, which also interact quite a lot with the sward structure and chemical composition that Mindy is grazing or eating. That feeding motivation interacts with the rumen, and it has a very detailed rumen that represents even mechanistically the digestive outflow rate from the rumen. And in this particular case, it's quite important due to one of the questions that I asked Mindy about methane production. On the other hand, and you see on the right side of the screen, the module that Mindy has got in simple terms to simulate urination, drinking frequency, urination frequency, and nitrogen concentration by the urination event throughout the day. So this is just to present you a little bit of a rich picture of what the model is and can do. Mindy is a dairy cow, developed here in New Zealand. But because of Mindy is built from the ground up, if you like, and she can be literally initialized as any other ruminant. In this particular case, we initialize or transform Mindy from the conditions of a dairy cow in New Zealand to the condition of an Angus cow in Utah. So we initialize Mindy, now the beef cow, with almost 550 kilos, with a level of milk production about 8 kilos of milk solids per day. Sorry, no, not milk solids, 8 liters of milk per day. And we transform a little bit of the variables representing the morphology of the cow. So that's a very long story short. And what we did in this particular simulation model, simulation exercise, is simulate 25 scenarios of what we call the treatment, and that came from a factorial arrangement between the frequency, weekly, fortnightly, and monthly, and dietary proportions from 5 to 20% of the total forage allocation, right, of either sandfoin or bird food trefoil allocation. So the pastures that Mindy was grazing and allocated to was composed or based on a grass pasture and supplemented with that particular proportion of those particular legumes. And the data that we used was data that was sent to my postdoc by Dan and myself, representing the chemical composition of the grass-based diet and the chemical composition of those legumes. We simulated the tannin content of the legumes in the detailed chemical composition that Mindy used. by changing the arrangement of soluble protein and rumen undegradable protein according to the proportion of tannins in the particular legume. The other thing that we did as well, and also it may not be that relevant to what we're talking about, we simulated the particular sword structure or canopy structure of those particular forages and those particular islands that we simulate virtually, of course, of those legumes to reproduce the closest grazing conditions in that situation. As we know, Mindy simulates grazing behavior, and the behavior of grazing will lead to the eating and dry matter intake, and also has carryovers when she moved from one forage to the other. So that's why it was quite important simulate a sword structure as well as the chemical composition, detailed chemical composition of the islands, in this particular case of those two legumes. So we asked Mindy two main questions amongst other ones, and those questions are the ones that we want to, or the results of the answers of those questions, and the ones that we want to share in this meeting. So first of all, we ask, what is the total methane production in grams per day? And the other one was, okay, what is the methane yield, methane of grams of methane per kilo of dry matter intake? And the third one was, what is the cost in terms of methane production per increment in production when we offer the island as compared to the control? So as you can imagine, when we offer the island, milk production, live weight gain, body condition score increase as compared to the control of only grass. So in the x-axis, you have got the control on the left, and you can go, you can have different proportions of sign point from 5 to 20 towards the left, and from the centers towards the right, from 5% to 20%. And the different colors indicate the different frequency of allocation. The black color is an allocation is once a week. The dark gray color indicates an allocation of once every fortnight. And the gray color indicates the allocation of the island once a month. So always they have in total the same allocation of island, if you like, right? So what you can see there, Mindy predicted different amount of methane production depending on what legume she faced in the island, but also depending on what frequency of allocation. So you got one of the most important thing is quite clear there. Mindy, depending on the frequency of allocation, change the methane yield. It seems like less frequent allocation increase the methane yield. But discussing with Juan in this result about these results, we said, well, yeah, that's something that you can predict there. That's something that doesn't surprise us. But if allocating legumes in this island have a increase in production, what does it mean? What does it mean in terms of environmental cost? Because the more the animal eat, the more the animal is going to ferment in the rumen, and the more methane we are going to have. We are going to produce. But in terms of the control, what is the cost of that increment of production in this particular case in methane? So as you can see there, there is at the bottom part, there is a different effect of legume and a different effect of frequency of allocation. So it looked like that when we allocate less or a smaller proportion of legumes, the cost of methane production per unit or per kilo of light weight and milk yield increase. As we increase the proportion of legumes in the diet, in terms of the island, the cost of production or the increments in production in terms of methane is reduced. And it seems like that the benefit gets even greater when we have lower frequencies of island allocation. All of that started to indicate that we can go for a really simple grazing management with greater proportions of legumes in the diet and lower frequency of allocations of those islands in the diet. So that could represent a relatively easier grazing management or management of the grazing pattern through the foodscape for the rancher of the grazer. So now we asked Mindy about the urinary nitrogen excretion. Here, we know that urinary nitrogen excretion, it's, if not, at least in our conditions in New Zealand, it's a greater pollutant as compared to methane because of the warming effect. So, and the more nitrogen the animals eat, the greater nitrogen the animal excrete. You know, roughly the efficiency or inefficiency of a ruminant and the general conditions, if you like, or temperate pastures go of 30%. That means that from all the nitrogen that is consumed, only 30% is translated into product, while 70% is excreted. And from that excretion of nitrogen, about 82% is excreted in urine. And the urine patch has roughly a concentration of 800 kilos of urea applied on the pasture. Imagine the plant can't absorb all of that, and a lot of it leach down to the waterways, and some of that became through evaporation end up being in nitrous oxide, right? Ammonia and nitrous oxides. But looking at the graphs there, you see again from the left to the right, you got control. And from the left to the right, we started with the proportions of sainfoin from 5 to 20% in the diet. From the center to the right, birch trefoil from 5 to 20% of the diet. The black color is weekly island allocation. The gray color is fortnightly island allocation. And the light gray color is monthly island allocation. That means that the cows face the particular island of legume for 2.5 days once a month, as compared to few hours every week. As you can see there, the more legume Mindy was consuming, the more nitrogen Mindy was excreting. And that's what one would expect, because not only total intake increase, but total nitrogen intake increase. Interestingly, at the same nitrogen allocation, if you like, at the same legume allocation, the lower frequency of allocation in terms of the island led Mindy to excrete less nitrogen. The least frequency was once a month. So, and again, that's an exciting result because literally simplified the grazing management at the field level, or they simplify the work of a rancher or a grazer to move animals more frequently to the island from one time to the other. But in here again, we thought, okay, well, we know that when Mindy was telling us the more legume she was eating, the more she's going to produce, but the more nitrogen was going to excrete. And we know, based on the previous graph, and the graph that is on top, that seems like the least frequency allocation should be the better. But what is the cost in terms of environmental impact of allocating those islands, as compared to the control? So we can see there that smaller or lower proportions of legumes in the diet leads to a greater cost per unit of increase of productivity in terms of live weight and milk yield as compared to the control. As we increase the legume content of the diet from 5 to 20%, the cost of urinary nitrogen excretion is reduced drastically. Moreover, and it seems like, especially when we look at sign point, that the cost is even further reduced when we have those islands allocated with the lowest frequency, right? And that's quite clear in Signpoint, and that was quite clear for the previous results in methane production as well. So the bottom line of this, and these are pretty much preliminary discussions we are having, is that yes, allocating legumes in forms or shapes of islands to beef cattle in a condition of Utah grass-based rangelands have benefits. No doubts about productivity. It can have an impact when you combine it with a proportion of legume in the diet, of size of the island, of frequency of allocation of the island, and the environmental impact. And the reduction of environmental impact seems to be the greatest when we allocate more legumes at the lowest frequency. So that's the part modeling one. I'm open to questions about that, and then we can tell the audience what we are doing in terms of design of one of our dairy farms and what the postdoctoral students or whoever is coming here can work with us or get trained or just see what we are doing from a design point of view and combining these concepts as well. You still hear me? Can you hear me, Juan?
Yes.
Okay.
So Pablo, I'm Jennifer, and I'm just curious about the increase in I think you said the composition of legumes. Is that equivalent to also increasing the concentration of tannins in the diet?
Well, first of all, hi, Jennifer. It's good to hear from you and see you in the audience. It's been a while. Yes, yes for both things. What we did is we simulated or we initialized the chemical composition of the legume, and then if you allocate more of that legume, the cow or Mindy had the chance to eat more of that legume. And definitely she ate more of that as well. So the greater allocation of legumes, the greater amount of tannins she will be ingesting, or she ingested in the simulation as well.
Thanks. Just the the other question is, do you know the reason why less frequency turns out to be less nitrogen excreted?
That's a very good question, and I haven't thought about that. A former student of mine did wrote a review article about frequency of supplementation, and he found similar results with the and different frequency of supplementation in low quality forest diets. I think what we are seeing here, so I should have clarified, but the results that that we seen here is the average of a month, right? So I really think that, well, to be honest with you, I haven't thought about that in detail. I think it's related to the carryover effects of, coming from a grass, go for, and especially in the lowest frequency, 2.5, almost 3 days intensive grazing or consumption of those islands, and then to get out of that island. So the carryover effect from the grass and then from the island to the grass, it's what may have had the effect there. But it would be difficult to disentangle if we don't get into a more detailed modeling exercise or asking for more parameters, more variables, not parameters.
Hello. I just had a question with regards to Mindy. How does Mindy control for possible extraneous factors such as the gastrointestinal sensitivities of the dietary change? Do we have as accurate as possible results? Is this going to be more of a longitudinal accuracy? Can we have immediate conclusions when we're making a change like this?
Well, first of all, let's, I mean, what do you mean by sensitivity in the gastrointestinal tract?
So I just mean that if you change the diet, that there is a chance of sensitivity with regards to any sudden change for just about any creature. Like if you're used to a consistent diet and then we change it, there's a chance of gastrointestinal sensitivity, which could change like the amount of methane being produced, like increase the amount of gas that you have. I'm wondering how we're controlling for those factors.
But first of all, Mindy simulate those changes is very mechanistic, and one of the modules that in fact the modules that we created with Juan, Fred, and Mike Forbes, we incorporated the module of minimal total discomfort and post-ingestive feedback in Mindy. So whenever, so they not only help Mindy to make decisions about what to eat or not to eat, what she would rather eat or not, but also help Mindy simulate the carryover effect of changing from one diet to the other. And Mindy can even simulate acidosis or those sort of things. And that's why I think it could have been Mindy could simulate those carryovers effects, from if she's coming from a grass-based diet, and getting into a legume, or from a legume to a grass-based diet again. But I don't think in this situation would have been quite significant or changing one diet to the other. And looking at the literature about, for example, rumen adaptation, there is still not consistency about how long does a rumen takes to adapt from one diet to the other. It could have been the case in the fortnightly or monthly allocation, but at the time when you look at weekly or even within the day, animals change diet from one patch to the other or from one meal to the other. And we have run some experiments even at the fortnightly level. If changing the diet from at the meal level shouldn't be a big effect there, especially with pasture-based diet. But when you go for a lowest frequency of allocation, and that's why your question is really, really pertinent. There could be some effects there, but in this particular case, those effects could have been beneficial rather than negative. You know, we've seen some of the effects that you mentioned when you go from grass to alfalfa, the high quality grass to high quality alfalfa, or even where I come from in Argentina, when we go from a Dallas grass diet to a lotus diet as well. There is a kind of stagnant period of about 5 days where the animals don't gain, don't lose weight. But then it seems like when they start regaining or adapting to the diet, there is a kind of uncompensatory effect there. So, but you know, let's keep in mind that this is, although this is a really sophisticated and detailed models that has 4,000 variables, those sort of things would be hard to catch even in the model.
Okay. Pablo. Yeah, so let's move on now on the visualization, some of the work you're doing on that front, and then that we could incorporate into this project on how these landscapes are going to be looking like.
Fantastic one, thanks so much. And look, if any of you have more questions about the modeling exercise, please let me or Juan know. It's a very long story, so it took quite a bit to get these two graphs. It's not that easy as it looked, and I'm more than happy to be in contact or just to have a Zoom meeting or to provide more details. And hopefully in the discussions that are coming, these results can be discussed, and so we can literally finish the paper and with a nice discussion. Okay.
And the brainstorming sessions, we can continue on that. What is the best way to graze those islands? Essentially, that'd be the question.
Yeah. So the other part that we're interacting with you guys here is in the terms of the designs and visuals. So there is another member of my team, Dr. Shannon Davis. I don't know if she's online or not. She may have not gotten up that early, but she's a foodscape designer in the School of Landscape Architecture here at Lincoln University. I'm gonna tell you a story to provide you an example of what we are doing. So Juan, it would be great if you can play the video that I sent you to introduce this part, which is when Juan played the video, you see this it's a dairy farm of Lincoln University, and we took a quarter of that dairy farm, and we started to design a dairy farm that is called integral health, where we are considering health from the ground up. What we are saying is like health is only seen when you look at that particular piece of land through several lenses. The lens of the soil, the lens of the vegetation, either grazable or browsable, the health of the animal, the health properties of the milk, and all of that will be reflected in the health of the community. But Juan, let me know if you're playing the video.
Yeah, it's playing now.
Okay. So as you can see in the video, this is the render of an architect who is doing a PhD with me in spatial agroecology. So what you see there is different components of the dairy farm. You have different forages of strips with different colors, you know, from pink wildflowers in the races to different strips in the paddock to what we call strategic browsing sites that had the function of shade and shelter and provision of aromatic plants, exercising aromatherapy and ingestions of some particular plant secondary chemical compounds, browsing shrubs, trees under irrigation, which is quite a challenge. This is a dry environment as well. A shelter belt around the farm, and then we have other wintering block with the same situation. And nearby the wintering block, you'll see what we call the motel and the birding center. The motel is where the cows during wintering, remember this is a seasonal milking season. During wintering, those cows eat winter forages and can have access to that motel overnight where they can urinate, and we can collect the urine in there so it's not deposited onto the pasture. And the birding center will have, cows calving under shelter with warm water and with ad libitum feed. There are a lot of things going on there, but the other thing that you may have seen when you saw those cows in the milking platform browsing, it has what they call the mulu. The mulu is those browsing sites that have a kind of a eel shape. They are surrounded by wood chip pads, so the cow can go and rest there under the shade or protected from the wind. This is a very windy region in New Zealand, and so we provide comfort there. And when the cows are either resting or sleeping, as we know, they get up or they wake up, get up and urinate. So we are thinking of capturing at least 30% of the urination in those wood chips that will slow down the flow to the soil. They absorb and adsorb some of the urine and nitrogen, and whatever go through is catch by the roots of those plants, woody vegetation plants. And each of those woody vegetation plants, trees, shrubs, and aromatics have a particular function. And we have created 2 vademecums or guides of plants in terms of plant secondary chemical compounds and the effect on the environment, the animal, and the human health. So I can't wait to share that with you guys, and with especially knowing that Stefan is there with Stefan as well. But if the video finish, Juan, let me know so I can show some. I can go through my slides.
Yeah, it's finishing now.
So that's the video. Just that's one of the things that I think would be really useful. Would one of you come to learn or to interact with the guy that built that video? That's the platform of what is going to be the virtual reality representation of that farm. From an architectural point of view, you can see in the screen now, literally it's an architectural map of how and where the things are happening at the farm there. You can see in the center, this is where it's the milking shed. On the top right, you can see the wintering block. And then on the left, the milking platform. So that's literally,
it's finished now.
Okay. But then what we are trying to do is create more visuals. How do communicate these sort of things to farmers, to students from a primary school all the way to university, rather than this dry map, if you like, right, 2-dimensional. So the first aim was the animation that you've seen that became virtual reality. But the way of conveying those things, imagine trees will take a while to grow, and browse will take a while to grow and be browse. So we are getting into this sort of thing. So imagine that, I don't know if any of you have been in New Zealand, it's like in this particular region, dairy farming is, as I call it, a desert of ryegrass. And you see 3 colors, black and white and green. So what we've seen here is not only different colors, different shapes, and behind all of that, different biochemistry that has multiple functionality. So this can give you an appreciation of what is being implemented right now and how we can start visualizing things to get the people more involved there. If we talk about islands, why we locate the islands, in this particular case, the browsing islands, if you like. They are located in a direction that, relates to the prevailing winds and also the prevailing night water flows underground. So these islands, also the root of these islands are supposed to catch horizontal movement of nutrients that could have or could end up in a nearby lake. So that's a representation of what's going on. It's a very, very long story short. I could be talking for hours, as each little thing that you've seen there has a scientific rationale behind that. Even the colors of the cows that try to represent moving from a Frisian-based cows to a more double-purpose cow. This is another view of those islands, if you like. So you can see the pivot, the irrigation, but you see the browsable tree, the browsable browse, and somehow the aromatics there, and the cows standing on the wood chip of what we call the paddock mulu. What you see here, it's a vision or a view of how the wintering block would look like. Cows nowadays have only one winter block, and just following the steps of Juan and Fred talk about diversity and how we can biodiversify our diet in terms of biochemistry. You'll see that that not only have more colors, different biochemistry, and different browsable plants there, in addition to the shelter. And on front, you see on the right what would be the motel, where the cows would spend some time there, by choice or by actual movements of the cows, eventually by virtual fencing. And they will have access to smart feeders. We got 4 of them, and those smart feeders will be providing some particular bioactives to each particular cow, and for each particular cow to exercise some sort of fetal nutrition or fetal programming to reduce or to literally change tissue deposition from a dairy beef, but also to change some of literally we can think of a sort of emotions, and with Stefan van Vliet and Juan know the results, we've seen that providing biochemical heterogeneity to use have a positive impact on the cortisol concentration, cumulative cortisol concentration in wool of lambs. So it means that you could argue that even in utero, biochemical diversity has a positive effect by reducing potential stress. But that's it for me, Juan. So I'm open to questions. Hopefully I presented what you wanted, but this is a very, very long story short. So I can't wait for some of you come here, or I can't wait to the next year to be face to face with you and paint you the whole picture.
Great. So yeah, thank you, Pablo. That's any questions, comments? I thought that this could be a really good learning, teaching tool and also extension tool for working with ranchers and showing the potential for establishing some of these islands in the landscapes where they can actually visualize how this is going to be looking like.
Pablo, is there is there progress on electronic fencing since you're in New Zealand and it seems that that's more intensive to start with than we are?
Yeah, the problem is with the companies, but there is quite advanced in there. You know, the companies don't want to sell these virtual fencings to research institutes. They're afraid of that. I got my, we wanted to implement, there is a big company here, but which is, it's doing really well. It's called Folter. And I've seen that. In fact, on the next door farm of our farm, we got 800 collars working and they work really well. Now, whether that or the grain that those collars GPS work to move cows from one day to the other, it's fantastic. The things that it can do, let me tell you, is fantastic. Differentiate pasture allocation at the individual level, if you like, of mobs, individual cows, of mobs, of 1,000 cows. But I think the grain is not yet as detailed for us to use and say the cows, well, you cannot get into that island, if you like, because imagine if one cow get into that island and you got a mob of 800 cows, they're gonna destroy it. So we are fencing it temporarily, those cows. But yeah, there is quite a lot of advance there in the virtual fencing, and I think it's a really good thing to try. And you guys have there another group of entrepreneurs, Kiwis, that is called Vents. They are releasing some sort of virtual fencing, I think, in Montana. So it would be great to start implementing at the range level. I think it would be great. We are trialing other virtual fencing at our rangelands in the mountains, which is called the E-Shepherd, but it has a less defined or greater grain, if you like. So the things at the range level would be easier to implement as compared to a more complex systems that I just showed. But if you got a simpler system, which could be 1 or 2 species, or if you didn't have those islands, of browsing, having virtual fence would be really good. There is another company, No Fence, from Norway. Hopefully I'm answering your question, because I think it's gonna come, and I think it would be a really useful tool to have. The question that I have, but it's not questioning the actual methodology, which I think it would facilitate and would save a lot of time of farmers, especially dairy farm, is like the cows respond to a negative stimuli, right? It's either noise or electricity. And you can argue, well, an electric fence is a negative stimuli as well, right? But once they learn, that's it, rather than being stimulated to go to one place and constant changing condition based on negative stimuli of noise or short electric little shots there. But that's my own question, and that's why we question the applications of virtual fencing in this integral health dairy farm, because the point of that is health, right? So, and health by definition of WHO, which involve not only the absence but the presence of emotional conditions that represent positive well-being.
So yeah, thank you, Paulo. So now let's take a coffee break. Yeah, let's try to do it really quick, about 15 minutes, and then we'll move forward with the presentations, and then that's great, triggers, Paulo, for the discussion this afternoon.
Okay.
So then we'll keep. So still the coffee is in the works. So 10:20. So we still have 20 minutes. So yeah, we'll keep moving with the presentations. I guess the next one is mine on deploying some of these islands in the range. So yeah, the next set of slides. That yeah, that's so yeah, this is continuing with well, in theory, what Pablo was describing on how these islands will be looking like in the real life, in the actual landscape. This is yeah, next slide. This is not working. Next slide. So that's a representation. That's what we did in this plot in 50-acre pasture in Richmond. We did this spray the grass, smooth brome grass, twice in the, as I was mentioning, in the spring and in the fall. And then at the locations where Doug Ramsey mentioned on how those actual locations were selected in using GIS. Yeah, the next slide. So, and that's the design of the, within each island, we got like strips, developed strips of seeding in November with 5 different species, so cows can choose when they're grazing there. Is alfalfa, trefoil, which contains, well, alfalfa with terpenoids, trefoil with condensed tannins, forage kochia, that's another, the one that we've been discussing yesterday, and that's, yeah, the seeding, yeah, I didn't mention that the depth of the seed, the kochia was way shallower in the relative to the other species. And then sunfoin and small brunette, which all contain condensed tannins, different types, different chemicals. They're not the same. So this is going to be strips. Yeah, next slide. And then, so the questions I have here is, well, that how best proceed to spray the weeds in the spring, cost of, and then the best chemicals to use for, well, because I know we'll be dealing with a lot of weeds. Then the timing, it's been really challenging of when to seed. Like Jennifer mentioned a little bit, when we see the last fall, nothing came out. Now we try to seed in early November, and we had storms. Then we have frozen soil, snow, moisture, that then not only to get it with the machinery, but also the seeds getting stuck with the moisture in the field. So that's things that, so the time and timing, I think, for this is really critical when to do it. So we did this non-till seeding. I'd like to also discuss some of the benefits, some of the negatives. So if we had to repeat this in the spring, which would be the best, like we did last this spring, how or will be the best or the best approach, I guess. That's another question I have. And then, well, how to graze these plots is going to be like a challenge. We got from Pablo, some ideas of, well, potentially like, really short term, very low frequency and short term, but then, so how you perform the rotations. I think we're also visiting about virtual fences, electric fences, when the time and timing, those are things that we need to discuss. So the rotations, not only to increase the persistence, so try to get these islands to persist in time, but also to enhance the nutrition efficiency of the animal. Maybe, as I was with that modeling exercise, show that maybe low frequency of consumption of these islands. Maybe, having patches or paddocks that moving the animals once a month and then let them graze the rest of the month on rangeland. That may be another option that I was thinking, just having has paddocks with diverse legumes and forbs, and then allowing for grazing for a couple of days, and then for the rest of the month just grazing on range. But yeah, which would be the best, I guess, grazing scheme for these diverse legumes and forbs. Yeah, the next slide. And then the next slide for long-term topics, animal health. So some of these islands contain secondary compounds. And then Stefan was just left. We are starting to collaborate on looking at potential benefits of these secondary compounds, like immunity enhancing benefits, so the cows being healthier, just a preventive strategy, not like just a treatment strategy, but cows being healthier in the field. And also how that impacts their offspring. We'll be following their offspring through the feedlot, so how that is going to impact the quality of the product and the performance of the offspring in the feedlot. Welfare of the animals as well, with those bioactives. We'll be measuring cortisol in the hair of those animals and trying to assess how, and then maybe some behaviors as well to assess animals should be in better welfare when they have a diversity of bioactives and plants in the feed as opposed to just grass. Another topic that we're starting to discuss is looking at soil health, and so how that soil is going to respond to these islands in the short and long term. And then seed dispersal is another thing that one producer described on one of his paddocks. He had some alfalfa, and then seeing that alfalfa was getting dispersed in the field all over that paddock. So assessing that those legumes potentially spreading across the landscape, that's something that we also planning on monitoring, but that's something to discuss as well. I think that's so I think that's what I have for discussing this afternoon on all these topics. So, and then any questions or comments before the brainstorming sessions, or? Good. Yeah, that's a good point. So yeah, the question was on wildlife and the potential wildlife impacts and how, yeah, we keep those animals. Also, that you mean in terms of persistence or establishment and then persistence of those islands. Yeah. Yeah. Yeah, that's a concern for sure. That's a point that in terms of wildlife impact and particularly in the establishment of these pastures. Yeah, not only from even large ungulates all the way even to small, even small. We had issues. I remember in the past with even voles or even small mammals. So all the range of issues that you can have on this, and that's a concern for sure. One of the things I was just thinking the other day is like, it's like, but depends on the population of animals, that that dilution effect that some managers, when working on aspen, they tend to, dilute the impact, the herbivore impact by having multiple allotments, I guess, of aspen when they just cut the aspen for regeneration. So then the abundance of but that's an issue that depending on how many, the population of wildlife you have, that may work or may not. If you just spread the herbivore impact over more patches, you might get some dilution on the impact on individual islands. So that's one option, but then there should be a threshold where that the population is that high that you just then you can control even if you try to make that dilution is not. So that's something that I guess we need to discuss more into the brainstorm session, how how to prevent that. Yeah, prevent those issues. You know, we tried, you know, with aspen. I did some experience with aspen and trying to prevent aspen damage by elk. And then one of the options was to try to saturate the animals with secondary compounds present in aspen, in supplements and in blocks and in all that. So they're providing all what those animals may need in terms of minerals and nutrients into blocks. But in some areas, you know, the population in one place in Colorado was so high that the population was so high that didn't have an impact, even if you just provide alternatives or you try to dilute that impact, it's still too high. So in some cases, it's really difficult. Yeah. Any other questions? Yeah, that's Jennifer.
I like Pablo's comment about having something in the islands that would scare off animals from grazing until you wanted them, and then you could turn it off like a pretend predator, that sort of thing. It seems like it might be a more manageable situation than trying to keep them out, would be to scare them away until you could take it away. I don't know. It was just something I hadn't thought about before.
Try that, you know, but then there's the issue of habituation on those in different wildlife species. I don't know if Dave is here, where if you're aware of some of these issues on habituation, where you get you know, like some well noise or you know, that type of and I've seen even research where they try to use some, you know, fake predators and you know, eventually the animals end up, you know, habituating to. So it works in the short term, but then in the long term the habituation takes place. Yeah, there's some other, you know, like some aromas, you know, from like extracted from, say, coyote or wolf urine. Those are treatments that people have applied, you know, to, you know, for preventing tree damage. And I think that's some of these have been kind of more successful with it seems that the animals with aroma of predators don't get as habituated as with visuals. I don't know if Dave or
Juan, can I make a comment
on that, or Rick? Have you heard about any wildlife damage? And
can you hear me?
So any other, so that's I think that some aromas, I know that people working on control mostly on pine trees, and they've used some combinations of aromas that are effective, and habituation is not that, but that of course is going to be for an island or for a pasture. Is going to be something that, you know, in terms of economics, or I don't think may be doable. So I think you have to probably hope that the animals would have other alternatives available.
Juan, can you hear me?
Yeah, maybe in this.
Juan?
Yeah, if you want to read the. Oh, okay. Coming up, or. Yeah, so we can hear you. What's your question?
Oh, that's good. That's good, Juan. But I was making a comment. We don't have problems here with predators. We have small predators that attack the birds, and we are using artificial intelligence and images to on cameras to detect those predators. And when they get into a particular zone, they are attracted and killed. I don't know if you want to kill the actual wildlife there, but you can. In this case, we want to, but you can have. I can pass you in the details, and even the company that have cameras that use artificial intelligence to detect the actual predator and then send a response. Either, well, in this case, we just shoot them, but we can send noise or other condition response. So in this particular case, the camera will learn how to detect a deer versus a cow, and when the deer appear, they could create noises of barks or whatever that repel or put the deer away from the island. But that was my comment. Sorry, I didn't mean to interrupt.
That's a good point. Yeah, I didn't know that. So essentially, are you using that technology already, or is it already available?
We are using that technology already in the mountains. We got a massive invasion of possums that came from Australia, and they are attacking our native bird population and destroying some of the trees. And so what this company is doing is setting strategic cameras around those hot spots. And when they detect a possum, literally they have a bait that attract the possum, and then, well, they humanely put them down, if you like. But that same technology could be used to other things, so that artificial intelligence can learn to detect a particular invader of that particular piece of land and scare them out, like with noises or other negative stimuli there.
And you reduce the likelihood of habituation because it's really targeted to a species as opposed to just something that goes off every half an hour or way more monotonous. And so that's probably, you take care of the issue of habituation. Yeah, there's some comments here.
I think when, if you start thinking about, native ungulates on vast areas of range in the West, especially if you're thinking about cost, it's going to, if I understood some of the prior presentation, what in many ways, what was desirable were small, very nutrient dense patches, broadly scattered across the landscape, which if you've got if you're in areas with a lot of elk or deer, I think that's where you were coming from, or antelope too. It's like, that's the worst thing you could do if you want them to persist, because they could camp on them. Although I have seen sometimes, if you can get things established for a couple of years, I've seen alfalfa that just gets grazed. I can think of a couple of places up on Deseret, up on the mountain there, that are oil pads that have persisted for 30 years with but if you can get things established before they start grazing, and maybe you could do something like that with temporary fence, but it's going to be awkward with, because that's part of the landscape out there. You can't kill them. You can try to deter them. They're pretty, most deterrents only work for a little while, but temporary fencing might be an option but after you got things established, they could essentially handle season-long grazing. The only other thing I can think of is to try to put your supplements in places where, that are sort of big game winter range, so that you don't have high densities of animals until the dormant season, when it wouldn't make so much difference. Like you say, antelope, you can't really, it's tough to get away from them on a lot of the range. But there's just a couple of thoughts on native big game.
In relation to those cameras, it's work being done out of Arizona, ASU, and Montana, and it's mostly for conflict reduction, but they're the smart cameras that they're training them to recognize different wildlife or livestock and essentially send you a warning, so you can deploy a range rider resources if it's wolf or grizzly, but they could be trained to identify antelope or elk or whatever, and then tie it to like what do they call them? The rag boxes that are the noisemakers. So you can, it would trigger the noise or the lights or the commotion. So, yeah.
Yeah, and it's more. Yeah, I have a question over here.
I just wanted to, I like this discussion, but it's applicable if we were doing this at a landscape scale, then these issues are applicable. At this point, with the demonstration plots, it's more about protecting establishment and getting the plots in place, which may take some fencing. And then after that, you know, just seeing what animals are in that place. But I think what's being talked about right now is great for the next project. And once we go to some sort of landscape scale, and then we can talk about satiation of the foraging, the ungulates, or the predators or whatever. But at this point, with these demonstration plots, it's just getting it established and then seeing what wildlife are in place happen.
Yeah, that's a good point. I think to me, I think what as Dave and Rick mentioned is like, well, once they are established, so then I think they can tolerate way more herbivory than the critical period is going to be the establishment. So that's and then yeah, that really small scale we can still deal with, you know, with fencing. Then as we move up to large scales, that's so all these other, you know, things that we've been talking about will be really applicable. But I think it's good because we are discussing, you know, long-term goals and short-term goals. So I think that's great that we are, you know, both thinking from both ends. That's the spirit and idea of this discussion. You know, it's going to the whole process and thinking what's going to be happening in 2023 versus beyond that, right? So that's great. Yeah, any other discussion here?
Yeah, I'm curious on the example that was given with those plots. That seems like improved pasture that's now trying to have diversity reintegrated into it. Are the other plots or sites similar? Because I guess in terms of like opportunities for establishment, that seems very different than a rangeland site with maybe some already diversity in it, or even seed bank versus even like New Zealand dairy production. Very different system, different opportunity for diversity integration. So are the other plots, the test plots, similar to that? Yeah. Okay.
Yeah, so there's a couple of ranches where they are also deploying plots with the same species we are using on this. This is the more kind of the research facility, you know, like a research, but then, and then there's potentially a third one, right? Matt, we're planning on Matt's, yeah, you can mention a little bit on that.
So, you know, Eric Thacker would be here and could explain all this in good detail. I'm going to try to sit in for him. He's really sick right now. But yeah, they are putting these plots out on current grazable rangelands is the plan. And so we've got several sites throughout the state. I think that 3 at least throughout the state that we will be employing these more on a rangeland setting, not on a research plot like the one that he's discussing here in Richmond, is it? Yeah. Yeah. No, it's just a place where we have control and can do some things that we couldn't do on a rangeland site with a farmer or a rancher. So it's helping us to kind of model it, to show it kind of on a small scale, but then we'll be showing, doing other plots throughout the state on more of a large scale.
Now it's Angie, is she around there? So it's ready. Okay. So yeah, now we can break. How long? 15 minutes. Okay. Hello. Yeah, let's continue. We are a little bit behind, about a couple of hours. But so now Dave Dahlgren is going to talk about the potential for wildlife and pollinators. And then that's on the silence.
Thanks, Juan. I'll try to be quick here and make up a little bit of time. And I can do that. I don't have any data to present. This is all coming in the future. So if you go to the next slide. So we've, as we've setting this project up, we've talked about some of the different avian species that we might look at. The passerines would definitely be part of these demonstration plots, and our plots are in areas that we could pick up some passerine birds. I was just discussing with Juan, like up in Clarkston, we have some grasshopper sparrows up in those areas and other songbirds. The key for me is finding reference to those areas so that we have both where the plots are and some sort of more wildland site that would be a reference to those areas. We've talked about grouse, but I think that applies more to a larger scale. Once this type of concept could go into a larger rangeland context, then grouse may become applicable. But I don't think we're going to see sage grouse at Richmond anytime soon. But once we head there, we can talk about those transects. What's that? There are Huns there. You might have to stop me from shooting them all before, but there are Huns up there. Yeah. So, but we'll be doing spring surveys for birds. So if there are Huns, we should, pick up. They'll be calling, that kind of thing. Next slide. I think this is the pollinators are probably where we're going to get the most data and the most differences, potentially, especially with these initial plots. And so we're staying coordinated with the other folks who are doing garden plots and back east. And but as far as methods go, so that we're all on the same page. But we've talked about camera traps for pollinators, which I was surprised people can even do that, but they can. It's very expensive, but we're looking into that. The easy one is to put out plates with just water in them, and you have plates of different colors that you can see on that bottom right picture to capture the insects. And then we can also do vacuuming or sweep netting of the material. So we're getting ground-based insects, and we're also getting insects that are in the canopy. But we are probably more interested in pollinators than just any kind of arthropod. And again, we'd be looking at this in treatment areas or the plots and reference areas. Next. The last thing, and this wasn't actually part of the proposal, but it's just something that's kind of, as I've been thinking about this project coming up, is the small mammal side of things. These plots are big enough and in areas that small mammals should be using these areas, anywhere from our rodents to some of the lagomorphs in these areas. So that's something, whether we use camera traps or some kind of mark recapture, we'll be looking into that. And then the large ungulates, again, going back to the stuff that we were talking about just before the break. In one hand, the ungulates are something we just want to avoid during establishment and keep them out there just to get established. Once they're established, though, we might be interested in how large ungulates are using the area. And they could definitely be something that once this goes to scale, it could be a real detriment to the longevity of the plot. So things that we might want to look to in the future. Next. That's it. I told you to be short. Any questions?
Those water plates look like great birds.
Okay, so I never thought of them as bird attracting like bath fixtures. They'll be small enough maybe that, yeah. Yeah, there you go. We do plan on bringing a master. I don't know, me and Eric Thacker might combine some of our research and bring on a PhD that would cover both. But basically, establishment has to happen before I can do the wildlife stuff. So we'll be looking at things this coming summer and then starting a grad student for the following spring. Anything else? I told you I'd make up time one.
Moving to the extension session. So that's Matt. Yeah, so he's going to talk us about the grazing schools and other plans for extension. I guess that's. Yeah, go ahead. The microphone. Yeah, I don't think he's. Yeah, and Eric is kind of hasn't replied. He's sick. Oh, I thought he said he replied. Yeah. Oh, okay. So let's see the.
But we can go on mine.
Oh, so yeah. So you pass the slides. Let's pass.
Yeah, those.
Oh, you're going to get those?
Yes, let's do those.
Okay. Great.
So, as I mentioned, Eric's not here, and I'm a poor excuse for Eric, so I'm not going to do what he could do. But I'll just mention a couple of things. Go ahead in the next slide. So he's working on, and I think he started establishing 4 demonstration sites, and these demonstration sites are on farms and ranches that have improved rangelands, but they may have only improved with grasses. So if there's a really good rangeland already, they're not improving those, putting these in those sites. They already have legumes and forbs and everything. He was really striving to find sites where they had maybe a crested wheatgrass or another established grass that was there. It's been improved, but we want to add the biodiversity in the forbs and legumes into those sites. And so he's working with ranchers to do that, put those in, and be able to implement these on a larger scale. Next slide. And the key is to implement them on a ranch site, and us in extension know how difficult that is. There's an art and there's a science, and we're really good at the science and the farmers are really good at the art, and somehow we got to bring those together to actually function on a landscape in an operation. So Eric's working with these sites to do that. They should doing some will be no-tilled, drilled in, and some will be tilled in. Next slide. So, go back one. So one will be in the Richmond one you already know about. Millville, Utah will be in the spring 2023. You see that Park Valley, spring of 2023, and Mount Pleasant will be a fall 2023. Those will be planted, developed, and ready to start looking at the research behind that and implementing it on our ranch scale. Now, I'm not Eric, but you can ask a few questions. And I'm sure he's got his email there. If you got a question, just email him. I've got his cell number if you want to text him. He's not well, so I wouldn't push him too hard right now, but in a week or two, he'll be back and going. If there's no questions, I'll keep her going. We can go to my slides now. So we in Extension already have some programs going for grazing and rangeland programs to educate producers. We've had these for several years. And so I was talking to Juan, I said, we can incorporate our current programming into this project. And that's where we are looking at here. We do a Sustainable Grazing Institute every year to teach intensive management grazing to producers. And again, we're trying to merge the science with the art. And the only way to do that is hands-on. So in this program, we have a day where we're in the classroom teaching them about the science behind this. And then we go out on site, as you can see in the pictures here, where they are implementing it. We give them a set of cows and a set of sheep, and they are to graze them for those 3 days. So we give them assignments. How big an area do you want for this amount of animals? How tight are you going to graze it down to? Anyway, we go through several scenarios so that it's a hands-on kind of approach with this program. We plan on, as things develop, incorporating what we learned from this project into some of the science that we're going to teach at this. Go to the next slide. And then we also have what we call the Central Utah Grazing Expo. This is, again, we're trying to get the art and the science. So we'll have a day of in-depth workshop for land managers, giving them critical land management information. And then we incorporate that with a tour. Again, we want to see it on the ground, not just in pictures. And so we incorporate a tour with that. So we'll go out, and what we've been discussing is actually on the landscape level. So producers can see, understand what changes are going on, and ask questions and figure this out. Again, this Grazing Expo is an awesome opportunity. As we develop these sites, we'll be able to go and tour them with this program. They're also looking at expanding this to Northern Utah, this type of a program, and be able to go right to the Richmond site and view that as well. So we're incorporating what we're learning here and getting it out to the producers we're already educating in this area. With that, I'll take any questions or comments about it. Yes.
Hello, so my name is Zubair. We are social scientists. I'm working with Jessica. I was wondering, when you say art and how your art is different from science or how it works, like what do you precisely mean by art? And when you are connecting with the ranchers, then what do you mean actually by that?
Yeah, so there are so many variables, but that a rancher or a farmer is dealing with to maintain the environment, maintain their productivity so they can still live on the land. So they've got to have a profit, and they've got to maintain the social aspect. And so they have all these variables. So when we bring a science to them, they have to figure out how they're going to take that science and put it into their current production practices to improve that. It's a very complicated thing to do. We look at it and say, oh, this is science, just put it on your farm. And they're going, okay, wait a minute. I've got wildlife. I've got fencing to consider. I've got my profit to consider. I've got all these other things. And so when we say art, we help them take the science and figure out how it's going to work to improve their farm. So it's a process. That's the art process. They're good at art. They know how to implement things on their farm. And so we have to find a way to educate them on the science and help them figure out how it's going to implement on the ground so that they can maintain a livelihood, improve the environment. Does that make sense to you? So that's the concept we're talking about with the art and the science.
Matt, thank you. What kind of producers would you say attend the workshops that that you've held, the Sustainable Grazing one and then the Central Utah Grazing Expo? Is there, some sort of characteristic of these people that typically attend them?
Yes, that's a good question. Most of the grazing expo is a lot of land managers. So this will be BLM, NRCS, and then larger ranches that are running maybe, hundreds to thousands of acres of land. So this is kind of more of the big landscape level group in that. The intensive management grazing program or our grazing institute is more of the smaller scale producers. These are, I get some larger producers, but most of it's the smaller scale producers that are trying to produce a lot of animals in a smaller area. So they're more the 100, 2 acres to 100 acre kind of group. So yeah, I'd say large landscapes one, more of a small landscapes are the other one.
Are the, I don't know if this is working or not.
Yep.
People in the small group, are they doing a lot of irrigated land?
Yes, mostly irrigated land. I get a few rangeland and we try to extrapolate it, but we're focused on intensive irrigated lands with that program.
Great, Matt. Yeah, thank you. And then, of course, we can continue on the brainstorming sessions in the afternoon. That's great. And then, so we have our next speaker is Jessica Shad, our social scientist, and trying to make sense of everything, how we are going to apply this, and does this make any sense? So
Yeah.
go ahead, Jessica.
All right. Hello, can you hear me on here? Okay. Well, yes, thanks for the introduction one. I'm Jessica Shad. I am a rural and natural resource sociologist. So I am over in the sociology and anthropology department. This is my student, Zubair Barkat. He's in his first year as a PhD student working solely on this project. So very excited to have him helping out. And we are working on task 6, which is focused on, what do ranchers think about what's being proposed through this project. And so as the project moves along through the 5 years, and we're able to share different aspects of the project, some more details, some of the visualizations or scenarios, we'll be going back and forth with producers, different types of producers, hopefully. Mostly here in Utah, but we do intend to expand it to the broader Intermountain West later on, so that we can feed back some of that information that we get from producers on their perceptions of what we're doing and how it might work in their operation. As Matt was saying, it's certainly an art and a science. And so talking to them and getting feedback from them about what sort of information they might need from us, how this might actually work on the ground. So what we're currently, can you go to the next slide, please? What we're currently working on, and sorry, I tried to squeeze this all into one slide. So as I just said, we're trying to get feedback from Utah ranchers on their own and broader potential uses of smart foodscapes so that we can inform both extension and outreach for the project. So this is a very iterative process, and I'm going to be sharing today one way that we are gathering information from producers currently. We will be using some other methods as the project progresses to get different types of information from them. But basically what we're currently doing is a very short survey on Qualtrics. How many of you love taking surveys? Come on. Well, so I teach a survey methods class to graduate students, and I'll be teaching it in the spring, but we're applying a lot of best practices to doing survey research. We know that producers are busy. It's not their favorite thing to be taking a survey from university researchers. They want to know how this information is going to be used. So we did develop a very short, in my opinion, survey that we're deploying currently on Qualtrics. So what we are having them do is watch the short 4-minute introductory video that if you were here last night, you were able to watch. If you haven't watched it yet, this is a great video that we've put together with some support that Kathy was able to offer to create this great short 4-minute video that introduces the project. And so what we're doing is we're having the producers that we send the survey to first watch that and then just give us some feedback. So 4 minutes of their time is asking them to view this video online. And then we're asking them a couple of screening questions. So are they currently ranching? Are they 18 and over? Do they have any livestock? And then we're asking them a simple question about what do you see as some of the benefits potentially to what we're doing in this project? What do you see as some of the challenges? We also ask about their willingness to try this on their own operation. And then we do ask a couple of background questions as well. So we only opened this survey up last week, or I guess 2 weeks ago. So this is just very, very, very preliminary data. I'm sort of embarrassed to even show this. We have 7 people who have completed it. So obviously we are going to be looking to get quite a few more responses from people. I will stress that right now we're just sort of looking to get producers or ranchers in the state of Utah to take it. We're less concerned about the representativeness of this data. Later on in the project, we'll be doing a mail and online survey where we're actually more concerned with trying to reach as many producers as we can. There were a wide range of respondent characteristics so far. Quite a few of the people who also went online to take it were ineligible. But what we've found so far is that there's mostly a positive overall view of the project after they watch the video. So 38% said that they have a very positive view of the video or of the project after watching the video, and none had a very negative response. Of course, there's some bias here based on who we've sent it out to, which I'll tell you about in a minute. Oh, I forgot to mention who we sent it out to. So in terms of the methods, what we've done is we were able to get email lists from the 2 extension workshops that Matt just mentioned. So we sent it out to attendees of those. And then I'm not sure if Eric has had a chance, given his illness, to send it out to a couple of listservs. So the Utah Rangeland Extension listserv, but also the Utah Cattlemen's Association. We're planning on sending it out to both of those. And then I know Juan sent it to those on the advisory board. So that's so far how it's been distributed. We are looking into purchasing a sample from a private vendor. So I'm working with them to try and get a list of emails of those who graze livestock in the state of Utah. So that should also help us reach some more producers to get their input. We also are offering an incentive. So if they take the survey, they get put in for a drawing to win $100 gift card. As I told you, I'm sort of obsessed with survey methodology, and I will tell you that's not the best practice to encourage people to participate in a survey. Does anybody know what's a good practice besides having a short survey? Yes. Yeah, they can go to spam sometimes. It depends on the population. Yeah, like we've done them before with USU students and gotten really high response rates. If you give a good incentive, yeah. But one thing that we often do, and I will be doing this in the survey that we do in a couple of years, is you give somebody something ahead of time. So I often send out $2 bills. So you send it in the mail to somebody and then they say, wow, somebody sent something to me and I feel like I should give something back. So when we do the bigger, more representative surveys later on, we'll be offering that sort of what we call to be a pre-incentive. So we'll see how this $100 ends up working out. This drawing for $100 ends up working out. So far, I mean, I said we've only had 7 completes. So sorry. People generally agree with the benefits that have sort of been outlined in the video. So they talk about perhaps the nutritional value for their livestock, eventually the decreased costs that might result in having different alternative sources for forage. Sorry, I just want to look, I had a couple of numbers. And then they did also note some concerns that we didn't specifically mention in the video. So initial costs to establish, cattle overuse of these plots. And then there were also questions about whether or not this only applies to cattle or also other types of livestock. So once I have more responses, I'll be putting this sort of information into a report to share with everyone. As I mentioned, we also asked about willingness to try. So responses to this range from 5 to 10. The average was a 7.6, or what I've classified as lukewarm. So nobody said that they were not at all willing to try. So as you can see here, for neutral was about 28.6%. So those in the middle who responded anywhere from 4 to 6 out of 10. Lukewarm, which was the most common response. So those who are willing, but not extremely willing to try, those were 7 to 8 out of 10. And then supporters, I classified as people who said that they were 9 out of 10 on the scale willing to try. So it'll be interesting to see when we get more responses. And then we also asked about different factors and how those might influence their willingness to try. And the most important was monetary compensation. So 63% that it said it was extremely important. Also, detailed information was something that a lot of people said important. So 50% said extremely important. And then I didn't put it up on here, but demonstration sites were also something that people are looking to see when they're thinking about whether or not this is something that they'd be willing to try in their operation. I also asked about knowing others who are using the practice, which is within some of my other research that I've done on other conservation practices. Farmers or producers like to see or know other people that they can talk to doing those, and that was the least important. So as I mentioned, next steps are really broader distribution of the survey. We do have an option for people to let us know if they'd like to do a follow-up interview, which some people have indicated that. So we'll be doing follow-up interviews and then later on a bigger survey. So one thing that I'm just looking for help with is distribution at this stage. So if you know people who might be willing to take this, forward them the email. If you have email lists of organizations that you'd be willing to share, I promise I won't bug people too much. And we're really looking forward to getting more input this way so that we can share it back with the team. Thank you. Any questions? Yeah. Do you do survey?
It's just, they don't respond.
Yeah.
Logically, like they're just, like we put it on Facebook, we put it on email, and we had like 42 responses, and we've sent out mailings now, like 160 mailings, and hoping we get a better response from our mailings.
Yeah. Did you mean you've sent something in the snail mail?
Yeah.
Yeah, so-
a stamped envelope, like.
Yeah, that's still the best way. So I've done a lot of survey research with different types of agricultural producers, and I would never, ever advocate for trying to gather data only online. Producers definitely respond a lot better to getting something in the mail and having the option to go online if they want, which some do, or filling out the paper copy. One thing that we were sort of excited about though with this survey was having the visual media, so the video. And so we were hoping that that would entice some people and really show them what we're trying to do with the project.
I was just showing one. It's kind of interesting just from what we have back. We did a livestock portion and ranked from the most important to least important. And number 1 was alternative feeds.
Okay.
And then in the rangeland stuff, the number 1 most requested was supplementation on rangelands. So.
Okay. Interesting. Yeah. Yeah. So, and keeping the survey short, which I have a hard time limiting the number of questions. And I promise I really did keep this one short. So those are the important things to do, and we'll see what happens. We haven't really tried pushing it out yet. So you'll probably be hearing from us, some of you that I've chatted with, to try and help us get some more responses.
Sure. Go ahead.
I guess this is for Matt as well. You'd asked kind of the user group that you're looking at. Are you looking demographics wise differences? I'm guessing older ranchers or their younger group? Is there demographic differences in acceptance that you're seeing?
Well, that's one thing we would like to know. So along with some of those opinion questions about the project, what they see, whether they'd be willing to implement, I think, just 4 background and operational questions. So one of them is age, one of them's education, one of them's acres grazed, and one of them is gross operation sales. So we tried to include a couple of those so that, yeah, if I get enough responses, I can break them out into different groups.
But I think that's a good point, is the age of the producer. The younger ones are usually more willing to try something new. The older generation has kind of figured out what they know works and they want to stick with it. And so there is a difference there. But I would just suggest old ranchers will talk your ear off. So if you go that direction, I would suggest you do a phone interview. That might be better than anything else, my experience with older producers.
Yeah, certainly. Yeah, this is just sort of a first stage, get some data at this point, and then, do some follow-up interviews with people. Yeah, those can be really long, but also really illuminating. Yeah. Yeah. Oh. Yep. Yeah. Yeah. I think one more question back there.
I was just going to say that if you were looking for ideas on how to reach out, printing off, even if it is a video, just like printing off the cards though, and putting them at like IFA, like supply stations where they're going to go anyways, and they're pissed off about how much feed costs. So you're more likely to get them to respond, especially when they're like, I don't want to pay for this. And then they look and see, oh, look at that. There might be another option. You're more likely to see some people, I think, that would be willing to fill it out.
Okay, great. Thank you. All right, I think it's education folks' turn. Yeah, all right.
Yeah, Kathy, so we are right on time now, amazingly. So it's so we move to our education component. Yes, so you, Kathy, run the show now. I think it's great to have you, and it's been great interaction. And I think this is a critical component of the project I was mentioning in my first in that introductory presentation, that education, all the reasons, well, at least one of the reasons on my story why education is so important. So go ahead.
Okay, thank you, Juan. Big shoes to step into following research and extension. And our slides. There we go. So we're the last 3 slides, I think. Well, I am happy to be here representing a larger education team. We have faculty members and collaborators from The Ohio State University. He is the geologist who works on our project. We have 2 faculty members from the University of North Carolina at Pembroke, which is the only university founded by Indigenous people, for Indigenous people, and on Indigenous lands. And that is our one of our science educators. And she also is in natural resources and environmental science. And then our entomologist on the project. We have 3 co-PIs here. I'm Kathy Cabe Trundle in science education, and my esteemed colleague and an endowed chair in arts education, Aurora Villa, will speak to you next. And then Catherine Vella, who is at USU Eastern. She is a mathematics educator, and she couldn't make it because of the weather, so I'll do her slide. But on our first slide, we wanted to share with you some of our successes. These are the things that we persisted with and the progress that we made on the project. And we are happy to report that, at least for the moment, we are on schedule with our timeline. We accomplished our year 1 goals, and we're well into our year 2 goals. Our first accomplishment was to write and we co-wrote a curriculum for elementary education. It's a STEAM curriculum, and the elementary goes from kindergarten to grade 6. And we wrote the curriculum with classroom teachers who are our teacher partners. And we have our education team working on it and a psychometrician's working with us from Miami University. And that's the one in Oxford, Ohio. But with those lessons, we're co-writing them with classroom teachers, and then those teachers will implement them, and we will gather information data-wise. We've got instruments that I'll talk about next. We gathered pre-data during this semester. After we implement the curriculum, we'll gather data again to look at the efficacy of that curriculum on content knowledge, on attitudes and behaviors that deal with biodiversity and sustainability. So the elementary lessons have been co-written, and then the teachers do a peer review process with that and give us feedback. So we have those established. During this second year, we'll have the scientist on the research team for the project. So Juan, Jennifer, et cetera, will give us feedback on those lessons. But our scientists within the education team have also written those lessons and given feedback. The second exciting development was to get our elementary gardens installed. And Aurora will talk about some of those challenges that we had with that, but we have those installed. Each school has 6 raised beds, one of which will include the smart Foodscapes, legumes, and forbs. One will include native wildflowers, and the others, the children plan on what they want to plant in the garden. And they may have more than 6 beds, but they have a minimum of 6 raised beds. They also have a greenhouse and a storage shed. So those are all set up. We also purchase curriculum materials, including children's books. We've gathered the elementary baseline data by doing the assessment of their content knowledge, particularly as it relates to pollinators and native pollinators, native bees in particular. We've looked at their attitudes towards sustainability and biodiversity, and we looked at it specifically for pollinators, but in general also. And then we looked at, so we did attitudes, behaviors, and content knowledge. So those are our 3 areas. We established a prototype for our education website, which will be linked through the website with Smart Foodscapes overall. And we began development on teacher professional development modules. It's not enough to just have the curriculum. There are lots of curricula out there. There are thousands of curricula. So we want to have the support for teachers. Our curriculum involves the integration of art into the STEAM disciplines, into the STEM disciplines for STEAM, which makes it unique. As you may know, we house Ag in the Classroom here at Utah State University, which is also USDA supported. A difference between what we're doing and what is already offered from Ag in the Classroom curriculum is that we specifically focused on integrating art. And also we are adding lessons that they don't have very many of, which is mathematics lessons. And we are also integrating citizen science. So we'll be doing pollinator citizen science and weather and soil citizen science. The last one that I'll mention, and this one is an exciting development, we are working with classroom teachers, as I mentioned, and we are disseminating with those classroom teachers. So we involve them in the research. So we right now, and in our first year, have published 3 journal articles with classroom teachers and some members of our team. And we published 1 chapter for an edited book. We have 3 other articles that are in review. We have 13 research presentations that are accepted to be presented beginning in January. So it will be a busy spring for us. And we have about 20 people who will be attending that first conference, providing outreach workshops with pollinator conservation and then citizen science. And then we have another workshop on climate change. And then we have research presentations there from our preliminary data. And If you notice the images of the children on the bottom, those are children from our middle school. We have our first middle school, and that one is in Far West, which is between Ogden and here on the way to Salt Lake. We have one elementary garden here on campus. That was the most challenging because if you're at a university, you know, universities have lots of rules and regulations. So it took from November until the following July when school was starting to get that approved, but we have it approved and we have the things installed. Our middle school garden came out of our seed grant, but they are currently partners in the USDA grant. And if you'll skip the next slide, I'm going to do Catherine Vela's slide for her. We wish she could be here, and then we'll come back to Aurora. So the plantings focus on the things that we plan to do that we have in progress right now. So the seeds that we're planting for the next part of year 2. We will have summer camps every summer for the next 4 years. This summer of 23 will be for elementary children, and it will focus on our school gardens and pollinators. So year 2, which is 2023, will be a pilot year for the elementary schools. And in 2024 will be the research year for those summer camps. So that's involving children and their families. During the academic year will be teachers and children. But as you know, a lot of the growing season and harvesting happens during the summer. So we'll be doing summer camps to maximize and do our data gathering with pollinators and citizen science in the summer. We'll have one camp here in Logan. And then I didn't mention the other garden for elementary is down at near Price, and it's at Wellington Elementary. And we'll have a summer camp down there in 2023. The research year is 2024 for elementary. 2025 is a pilot year for high school and middle school students and their families. And then in 2026, that we'll have our research year with the camp. So we already have 3 sets of gardens in the elementary and the middle school. We're starting with the middle school down near Price, and we're currently recruiting the high school partners. We'll have one high school partner here in the Cache Valley, we hope. We hope because of commuting can be a challenge. And then we hope to have another high school down near Price. We are working with Eric and the extension team to develop information sheets through USU Extension. We're involving our undergraduate research assistants and our graduate research assistants excuse me, on those information sheets. And those are targeted for school gardens and teachers and families. And those will be published through USU Extension and have the representation for this project. We also are working on a children's book. We have money in the budget to write a children's book. We are partnering with the Shroud Institute. It's a water institute. It's a nonprofit in Maryland. They wrote and published Creek Critters, and it's part of a citizen science project too. So we're going to do our children's book to focus on pollinators and native bees. And that will be a co-authored book between the education team, the extension folks, and the research team, our scientists on the research team for the project. And then we are doing the documentaries. The STEAM career documentaries will involve interviews with the same videographer who did our introductory video for the project. We are having children interview our scientists, and it's modeled after a project that I work with National Geographic, where National Geographic explorers explain their research with all their passion and enthusiasm to little children. So our scientists will be interviewed by classroom children in elementary and talk about what they're doing for this project. And that's important. Well, and then we'll have another set, one set for elementary and one set for secondary. So we'll involve little elementary children doing the interviews first, and then the middle school and high school students will do another set of interviews. Those are important because for children to be able to be interested in science and STEM careers, they need to have higher science capital. Part of science capital involves what you do, what you know, and what you read, and then also who you know. And by having the career videos, we hope that that will help them feel like they know the scientist who can present their work in an approachable way. We've already started on the middle school lessons, and we will start in January with having the teachers start the review process. And then Aurora is up next to talk about our challenges.
Thank you, Kathy. So hi, I'm Aurora Villas. So I'm a professional practice associate professor here at Utah State University and Beverly Taylor Sorensen Endowed Program Director for Elementary Arts Education. So I'm really excited to be involved in this program. My focus is more on arts education and arts integration. But I do want to talk a little bit about our patience that we've had to have during this project. Kathy talked about perseverance and progress. And as you know, there's lots of successes and there's lots of challenges that come along the way. So we've had a few challenges and things that we've needed to be very patient with. The first one I wanted to talk about is just the lack of availability of school garden materials or significant increases in material costs. As you know, I think since COVID, we've just had a lot of problems with getting things in, right? With building materials and whatnot. So prices changed, and we had definitely some delays in availability of different materials. High rate of department administrative and staff turnover. So in the School of Teacher Education and Leadership, we had a total revamping of the HR and the business managers. So that created some setbacks for us in getting some of the paperwork in and getting people paid and things like that. But we are now with, I think, a whole new team, and we're moving forward. Lack of available space in the College of Education and Human Services. Right, we don't have a lot of space for things to store supplies. We have so many supplies that have come in. I believe Kathy has some of them in her house right now. Different things that we've been able to purchase for the educators. So the educators are so fortunate to receive nature journals and stickers and water bottles and lots of children's books. But we have had a lack of storage space, but I believe the College of Natural Resources has been very generous in extension to provide some space for these boxes of supplies. We haven't had any office space for the graduate research assistants, but I think, again, College of Natural Resources and Extension has provided some space for that. And we have had some delays with facilities here at Utah State University. So those of you that have worked on university campuses, you know how it can be, right? With working with lots of different partners and collaborating with different people. So we have had some setbacks or delays in setting up the garden approval and installation here at Utah State University at Edith Bowen Lab School. But we're excited that we got it to work, and 8 months, I think, was it 8 months altogether? It took from request to approval to get all of the beds approved in the garden plots. But we're excited to move forward. And Edith Bowen is just a wonderful place to partner with, and the students will benefit from it. Kathy, am I missing anything?
I don't think so.
Okay.
I will say that for the other school districts, we met with them once or twice. With Utah State University, we met with them more than 30 times about the garden.
So Kathy mentioned that the other schools that we're partnering with, it was maybe once or twice we had to meet with them, but Utah State, we had to meet, what did you say, 30? More than 30 times with different facility meetings, and it was, yes, a long process. So I think at this point, we'll open it up to any questions that you might have for us. Jennifer.
Thanks. I just wondered what kinds of art projects you anticipate including for the children. We know about the gardens themselves. Is that the art, or are there many other kinds of arts that are going to be incorporated? Or is that even part of, I don't
No,
even
it's a
know.
great question. Thank you, Jennifer. So as far as in our breakout session today, we're also going to show some examples of some of the projects that some of the lesson plans. And one of mine is an arts integration lesson with star books, which show the life cycle or metamorphosis of insects. But to answer your question, yes, we have several lessons that we've created. 49, I don't know, there's so many. Now
Our goal was 30 and we have 49.
49 elementary lesson plans. Not every single one of them is arts integrated, but many of them are. I believe we have about 15 of those are arts integration lesson plans, and they integrate dance, drama, mostly visual art and music. And so the garden plots are just one piece of everything. And so many of the students will go out to the gardens. They'll actually draw flowers from the gardens. They'll do observational drawings. They'll create movement in response to some of the lessons. So I hope that answers your question. But yes, there's several arts integration lessons that we've developed.
And Aurora, tell them there are 3.
Come on up here.
I just wanted you to tell them about, you have a team of 3 art experts.
Yes.
And they cover different types of art.
Yes.
So if you'll mention about that,
Sure, I'd
because
love
it's not just about visual arts.
Yes. So I do have an arts team that's working with me. So I have Ashley Boyack, who is a dance educator. And so she's designing all of the movement dance integration lessons. And then I have Cindy Hall, who is a retired music teacher, and she's developing all the music integration lessons. And then Lisa Saunderson, who is the art teacher, Beverly Taylor Sorensen art teacher over at Edith Bowen Lab School. She is the visual arts expert. And then my background is in visual arts. So I'm writing a lot of the lesson plans as well. So yes, it has this wonderful focus in many of the art forms, not just visual art.
It's coming from an extension guy, but getting the schools to actually do the garden during the summer when our growing season is, they're usually out and not available. Have you had some struggles getting them to be okay with that and having someone to maintain those gardens through the summer?
I can have Kathy talk to that a little bit more.
We have, and that's a great question. We have 3 teachers at each school, and one of whom is a garden manager. So that person works with us every semester. So they work a 12-month contract with us. Well, it's not actually a contract. That's one of those HR issues. They have 12 months of participant support to work with us. And starting with our camps, we'll have the camps there during the summer to have the children working with them. A very wondrous and wonderful happening at the middle school. The middle school children formed a garden club with their garden. They meet every Wednesday afternoon, and they asked the teacher if they could meet throughout the summer. So this past summer, they met every Wednesday to continue their garden club. So the children initiated that. So, a lot of fun, wonderful things happen, but we did think about that. Rita and Caitlin, the 2 folks over at UNC Pembroke, they've been working with school gardens for about 20 years. So they've run bee camps before, and they helped us to identify some potential pitfalls, which is what's going to happen when the garden is most productive. How are we going to use that? So our lessons will be designed for the K-12 curriculum for public schools, but it's available to anyone online. And we see folks using this in informal science also, and homeschooling. So we'll be promoting it broadly. So hopefully it will be used by lots of folks during the summer. But thanks for the question.
Hello. It's not on. Is it? Okay. I'd love to hear a little bit more about the summer camps. How long are they? Are they going to be free? How are you going to advertise? Where are they going to be? I have some kids that I'd like to sign up.
Edith Bowen is one of the sites, but I'm going to let Kathy you want to talk a little more?
Yeah,
Yes,
parent
we'd love
involvement. We have, where they'll be building native bee homes. Well, and I don't know if you know this about Utah. If you notice my accent, I am not from around here. I hail from the great state of Tennessee, but I do have to tell you this about Utah. Utah has more biodiversity in native bees than any other state. And there are more native bee species in southern Utah than the entire eastern seaboard. So we have a quarter of all the native bee species in the United States are in Utah. So to educate families and children about the wonders of native bees in Utah and what they can do to modify landscaping or gardens, and then to invite native bees into their landscape with changes in planting, but also in terms of these native bee homes. So yes, we'll have one at Edith Bowen. It's a week-long camp and it's an all-day camp. And then we'll have one down at Price at Wellington Elementary. And then the middle school one for Cache Valley will be down at Walk West, which is in Far West. And then we'll have another one down in Price when we have our middle school, high school down there. So they're week-long camps. And as I mentioned earlier, our co-op PIs over at UNC Pembroke have quite a lot of experience with the summer camps. They actually piloted our lessons this past summer in their bee camps. So we already have some data on that we've been able to make revisions. So it's been fun and exciting.
But we love your parent involvement.
Yeah. We'll sign them up, Jessica. I thought about, we also are involving, we have about 3 preschools on campus, and to be able to install our gardens at Edith Bowen, we were asked to include them. So we've included them, and I thought about doing something for the preschool because I have preschool grandsons, but that little is a lot. You need one-on-one with them. Other questions?
Hello. So before coming here, I used to work as a monitoring and evaluation consultant for DFID. So I was wondering, like, how do you measure the success of all the activities or the initiatives you are taking, especially with the increased knowledge or any behavior change with the children? Like, do you measure it, or like you're just doing the activities and what's next?
No, then that's a great question. It is a research-based curriculum. We did, Rita Hakovic and I, my colleague over at UNC Pembroke, we did a lit review of the past 30 years of garden-based learning research. So it's everything that we've designed is based on that research, and also research on social emotional learning, which is more important now than ever, especially with the COVID pandemic and isolation with children. So it addresses the heart, the hands, and the mind. So it's hearts on, hands on, and minds on, heads on for the social emotional learning. But it's based on those 2 research bases in terms of what we designed, in terms of looking at the efficacy. I mentioned that we are working with a psychometrician, Dr. William Boone at Miami University in Oxford, Ohio. He was at Indiana University, and he has helped us. He spent the first year of the project vetting instruments, and a lot of them came out of Cornell and some of their work. So we are piloting the curriculum and piloting those instruments during year 2 with the elementary. And then we will be gathering data on the curriculum. The research year is year 3 for elementary. We pilot in year 3 for middle school and do research in year 4. We pilot the curriculum for high school in year 4 and do the research in year 5. But it's all based on, it's all research based on how we design it, how we revise it, etc.
Okay. Thank you.
Go to Salt Lake. No. Yeah, with this ice, man, it was.
So yeah, just keeping things for the meeting. So we are moving after lunch that we are having here to the breakout rooms in the Natural Resources Building, which is just across this road. So we are just steps from there. And then there's going to be two rooms, extension and education, which will be, NR 202 and 204, that they're side by side. Several of you, I think, already been there several times. And then the research brainstorming room is going to be by the dean's conference room that is downstairs, also like just down the stairs. So there's going to be plenty of opportunities for people to move across the rooms because they're right there. And then for people that are participating through Zoom, the reminder is that there's going to be a new link that you received last Friday with actual Zoom links for research extension and education. So that's, I'll try to open the links before we go to the meetings, and then so you can just zoom in anytime, and then also switch rooms like in a free choice. There's also going to be, like tomorrow after we finish at noon, a field trip really close to campus, which is the Forage Lab here by the parking lot. So it's really close to campus. And then they are growing some, they have some trials in the greenhouse. So it's going to be basically checking at some growing plants now in the winter. I guess that's the only spot that they won't, I hopefully won't have any snow. And then so if you let me know, we'll start building. I think, Angie, if you have a list of people that are willing to, or they have the time to stick around for this field trip, short field trip, shouldn't take more than an hour. So we see if we have enough critical mass of people that are not traveling and they're willing to go to this field trip. So we break the monotony of being in, well, instead of a room, we'll be in a greenhouse. So I don't think the change will be that significant. And I think that's pretty much it. So I know when lunch is going to be ready. So I know if you have any additional comments about before we break for lunch, anything of the topics we visited this morning. And then it's also going to be a chance for people participating through Zoom to in the chat room to ask questions or comments, and then we can maybe read those before we have lunch. And then actually lunch is going to be, what time is?
Supposed to be about 12:35, but I just asked her if they can go earlier, but she's checking in.
Okay, so around.
So if you want to do question things, then tell us.
Okay, so if you have any more comments or questions, that'll be a good time. And then I'll try to see in the chat room if there's any questions that we can bring up.
What time are we starting with the breakout?
Oh, it's right after lunch. Yeah, let me see what we have planned there. I think it's 12. So as we finish, yes, it's 1:40. So if we start at 12:35, yeah, so that'll be the right time. About 1:40, we just move as a group to that building.
And are we meeting it here tomorrow?
And then tomorrow morning we'll meet again here. So at 8 o'clock, which is going to be the same process. We have to pick up one person from each group where we can report basically a short summary of what we discussed in those rooms for people that didn't participate in that particular room. So we'll have reports essentially from each room, and then it's going to be just an open floor for discussion. That's, I think, yeah, like in this morning, I think it's been the most rewarding part of the meeting, I think, getting this type of questions and discussions. So I think that's next tomorrow morning is going to be like that until noon when we break for heading to that to the greenhouse. So that's the plan. So anymore, yeah, and let's see, I'm going to check if there's any questions on the chat room. Is there any people there? So are people logged in or you already? Yeah, so the people okay. So any more questions or comments? If not, we just can break out and continue with the visiting until lunch.